In this lab, you'll be studying the reaction that occurs during the browning of fruits and vegetables. This is the reaction between catechol and molecular oxygen to form water and 1,2-benzoquinone. Benzoquinone shows up in the visible spectrum, so we'll be using the UV visible spectrometer to monitor this reaction. When you get to lab, the first thing you should do is turn on the UV vis spectrometer. Then you'll want to go over to the computer and turn on the instrument online software. So when the dialog box prompting the operator name and password comes up, you can click Cancel and wait for the software to load. When it's loaded, go to Instrument and then click on Lamps, and make sure that both the deuterium lamp and the tungsten lamp are turned on, and click OK. You'll want to wait at least 8 minutes before you take a sample spectrum to let the lamps warm up. So next you'll need to prepare your catechol solution, which is the substrate for this reaction, and follow the instructions in your lab manual to do that. And then you'll need to prepare the apple enzyme solution. So peel the apple and then cut it up and put it in the blender with 200 mils of ice and water, and then blend that until it is smooth. Now that you've pureed the apple and ice water solution, we're ready to pour it. So we'll want to filter it through a cheesecloth. So just put the cheesecloth in a beaker, and then you'll want to pour this apple solution in there. And then it might not go through very easily, so you might have to kind of squeeze the cheesecloth. So just kind of make a ball and then like squeeze it until the puree comes out. And then pour this into an Erlenmeyer flask and take it over to the hood where you will keep it on ice and bubble nitrogen through it to reduce exposure to oxygen. You can put the pipette tip that's connected to the nitrogen line in the apple catecholase solution and then turn the nitrogen gas on to bubble nitrogen through the solution. And this will reduce the exposure to molecular oxygen and prevent our solution from browning. Now you need to set up the instrument to take the absorbance measurements at 540 nanometers. To do that, under the task menu, make sure it's set to fixed wavelengths and then click Setup. And then in Use Wavelengths, you want to type in 540 nanometers and select OK. And now you want to take the blank spectrum. So fill the cuvette with DI water and wipe the outsides with a Chem wipe to remove all fingerprints and oils. And then place it inside the sample compartment chamber and press the arm down to lock it into place. Okay, so to collect the blank spectrum, just click on the blank button and that will collect. Now we're ready to collect the data. To prepare the cuvette for the sample collection, you'll want to put the appropriate amounts of water and catechol in the cuvette. Have your partner get the apple enzyme solution from the hood, and then you can put that into the cuvette as well. Then put the cuvette into the sample chamber. But instead of just taking a sample, you'll need to wait 5 minutes for it to equilibrate to room temperature. So you can use a stopwatch to do that. So when you're ready to collect a sample and you've waited for 5 minutes for it to equilibrate, you can go ahead and click Sample to start collecting. And then you'll want to start the timer and collect a sample every 10 seconds for at least 600 seconds, or about 10 minutes. So after our stopwatch has gotten to 10 seconds, we'll want to collect another sample. Continue with this until you've got at least 50 scans. When you have all the data, you'll want to highlight all the rows and then copy and paste that into an Excel datasheet, which we'll use for the data workup part of lab. Repeat this process for the rest of the trials outlined in the lab manual. So in the second part of this experiment, you'll need to choose an inhibitor and then prepare 50 milliliters of that solution, and then use the same type of apple that you used on day 1 to prepare the enzyme catecholase solution. Collect the blank, and then collect kinetic data using the volume of substrate over the center of the linear portion of the Lineweaver-Burke plot. Then vary the concentration of the inhibitor by factors of 10. You'll need to make sure that you have different inhibitor concentrations so that you're able to monitor the progression of the reaction, which should be noticeably slower than the uninhibited rate. As you're monitoring the solution, make sure that you are collecting data over the appropriate range. To make sure that this is correct, go to Method and select Setup Analysis, and then display spectrum from 400 to 600 nanometers, and select OK. So after you've found the optimal concentration of the inhibitor, begin by varying the volume of the substrate to collect data necessary to create a Lineweaver-Burke plot. You should also prepare two solutions where the inhibitor concentrations are near the optimal one, but differ by a factor of 2 from one another. After you've completed the experiment, make sure that all the waste is properly disposed of in the appropriate containers. The apple puree can go down the sink, but everything else should be in the labeled waste. When you leave the lab, make sure that you turn off the UV-Vis spectrometer and log out of the computer.